Our Do Now today asks, what are antibiotics? And we have some good answers here. They fight off bacteria, they fight off bacterial infections, they treat bacterial infections, help you get better if you're sick, they destroy bad bacteria. And there we go, Kaylee, that's the answer I was looking for. So we want to make sure we are specific when we are saying these things, right? Like it fights bacteria. Well, antibiotics aren't actually like little creatures that prepare for battle and go against bacteria, right? They are a medicine. And specifically, they are a medicine that kills bacteria. Okay, so we want to be specific in that. I saw a lot in the antibiotic reading of they cure bacteria, they treat bacteria, they fight bacteria. But what does that really mean? What do they really do to the bacteria? Okay, so they kill the bacteria. So we are actually going to start here then. We're going to kind of review. And as we review and go over what you learned in your antibiotic reading, we're also going to update our evidence tracker. Now remember, that means every time we kind of discover some new information, I update it in this evidence tracker with you guys, and you have access to the modules and through the button in the home page in Canvas. That way, if you ever need to look back and reference like information we gathered, learned, or observed, you can always access it from this evidence tracker. Okay. So we want to kind of start by reviewing and making sure we understand the basics of that antibiotic reading. So the first one was what we just covered. Antibiotics kill bacteria. Okay. Now again, I stress they kill bacteria and they kill bacteria. They don't kill viruses. They aren't used for germs. Germs is this weird, generic, common language we created to describe things that make us sick. But germs could be parasites, they could be viruses or bacteria. And in this unit, we are specifically only talking about bacteria. So antibiotics kill bacteria, and they only kill bacteria. They can't be used to treat anything else. Okay. So what does it mean then if bacteria are resistant to antibiotics? What does it mean if bacteria are resistant to antibiotics? What does it mean to say that bacteria are resistant to antibiotics? Okay, the antibiotics won't affect the bacteria. Okay, so what does that mean? What won't the antibiotics do to the bacteria? You're correct. But what is it and what does that mean? When you say it won't fight it off, what does that mean? Again, antibiotics aren't little creatures like in battle against the bacteria. So when we say it won't work on the bacteria or it won't fight off the bacteria, what are we really saying? That the antibiotics won't do to the bacteria. We just went over the answer. Kill, yes. Because what do antibiotics do? They kill bacteria. So if bacteria are resistant, then these are bacteria that won't be killed by antibiotics. Okay. So that's why it was so serious and such a big deal in Addie's video when they showed that she had become pan resistant. Pan again was saying that every single type of possible antibiotic that had thus far been created by man would not kill the bacteria that were inside her body. Not a single one of them. Okay. Now, the reading then kind of wrapped up by kind of giving some tips of how we can kind of avoid or decrease the chance of bacteria becoming resistant. Okay. And so the main thing here is that we should only be using antibiotics when we have a bacterial infection. The meds are prescribed to us, and use the full dose. Okay. So that means, you know, when you get antibiotics and you're sick, not only taking them till you feel better and then stopping. That actually produces resistant bacteria. And eventually we'll talk about why and how exactly that happens. Only using antibiotics when we have an infection. You know, when we first realized that we found a substance that could kill bacteria, we were in love as a society, to be quite frank. You know, for hundreds of years, there were pandemics across the globe and people dying from antibiotic infections, or sorry, bacterial infections. And so to finally find a substance that could kill the organism that made all those people sick and die was super exciting. And it was so exciting that frankly, we started to put it in everything. We put antibiotics in our hand soaps. We put it in body washes, shampoos, face soaps. We put it in body lotion. We also put it in food. We spray it atop produce that we eat. We put it in the food that we feed to cows, pigs, and chickens so that when we eat those animals, they aren't full of bacteria or they have a lower percentage of bacteria. Everything we are constantly putting on our bodies and in our bodies is full of antibiotics that were not prescribed to us. We don't need because we don't have an infection. But we've put it in everything because we've almost become like bacteria phobic and afraid that if we don't put antibiotics in everything, then we're going to get sick. Okay. But actually, the best way to actually just kill bacteria is what? You do it all day, every day. How do you get rid of and kill the bacteria on your body every day? How do you get rid of and kill the bacteria on your body every day? Okay, and when you shower, what do you use? You just stand under the water? That's all you do? You stand under the water. Oh, soap. Soap. And it doesn't have to be any special type of soap. It doesn't have to have antibiotics and it doesn't have to say antibacterial. Plain soap is actually the most effective substance at killing bacteria. That along with, of course, like rubbing alcohol, but we're not always going to douse ourselves in rubbing alcohol. So the main ingredient of what we use daily that kills bacteria effectively is soap. Just like soap attaches to grease on our dishes and pans when we clean them, soap attaches to the cell membranes of bacteria and actually rips them open. So every time you wash your hands and every time you wash your face and your body, the best way you can guarantee that the bacteria that were on those surfaces are gone and killed is to just make sure you use lots of soap. And a lot of times people will also say, you know, what about hot water? You have to wash your hands with hot water. Well, actually, no, you don't. Because the temperature required to actually effectively kill most bacteria would be boiling. And we are not washing our hands with boiling water. So genuinely, the most effective thing you can do regularly to keep your hands and body free of bacteria is to use lots of soap. Okay. Right. Does anyone have any questions on anything that we just went over or anything that came from the antibiotic reading? Okay. So we went over the do now. We went over the antibiotic reading and questions. And again, remind yourselves that anytime you get a score on something you're not satisfied with, you can always revise, correct, and resubmit it. So that could mean you get a 1 or a 2, or even if you got a 3, maybe you know, like, oh, if I just do this simple, small fix, I can get a 4. Do it, okay? Easy to bring your grade up. We updated our evidence tracker, and again, you have access to this through your modules, but also there's a button on the home page. So if you're ever working on a homework assignment or a model and you need to refer back to stuff that we covered, you can always find that there. All right, someone asked a good question. They asked, how do I see your grades? So when you ask that question, do you mean your overall grade in a class or grades on assignments? So you cannot see overall grades in classes. That's why I send you guys emails every week to let you know what your current grade is. I can't really show you because it'll show everyone's grades, but I did send everyone an email last week that told you how to check scores on assignments and how to roughly estimate your overall grade in each class. So I would look for that email from you and read over it. Okay. All right, so we know bacteria can make us sick, but not all of them. We know that bacteria are kind of all over the place. And we know that they can be killed by antibiotics. And so if they can be killed by antibiotics, that means that bacteria are living organisms like you and me and the plants outside. So to fully understand bacteria, we need to remind ourselves that they can do the same living characteristics that we can do that we covered in the beginning of the school year. The bacteria are made up of a cell. They're just one cell, but it's still a cell. They respond to their environment just like we do. So they respond to temperature changes, changes in the amount of food or water they have, if there's chemicals like antibiotics in the surface. They grow and develop, and they also need food, water to survive, just like we do. Okay. Lastly, they reproduce, right? They have to make more of themselves, or else how did more bacteria get all over everything all the time and spread? And so we want to now learn about how do bacteria make more of themselves, right? We know that Addie picks them up and they entered through the scab she was picking at in her leg. But that wasn't like a million bacteria that she picked up. It was probably just a few. So how did Addie start with a few bacteria in a scab to a full-blown, full-body infection that could not be treated with any antibiotics? So we're going to watch this video here. It's going to talk about bacteria and how they reproduce and divide. We're going to watch it twice like we do for all our videos. And at the end of the video, we are going to click the reply button, and your job is to post, how do bacteria reproduce, or how do bacteria make more of themselves? Okay. So again, at the end of the second time watching it, you're going to click the reply button, and you are going to post, how do bacteria reproduce, or how do bacteria make more of themselves?
Prokaryotes are microscopic single-celled organisms, including bacteria and archaea. Prokaryotes are everywhere. If you looked, you'd find bacteria on every object around you. One reason bacteria are so ubiquitous is that they multiply very rapidly. How do they multiply so quickly? Let's look more closely at prokaryotic cell division. Prokaryotic cell division is a process by which a cell divides into two or more cells. The process of prokaryotic cell division is called binary fission, which is a simpler process than found in eukaryotes. Recall that prokaryotes differ from eukaryotes due to the lack of a nucleus or cell organelles. Typically, their chromosomes consist of a single loop or circle of DNA. The circular DNA molecule replicates. It then attaches each copy to a different part of the cell membrane. The cell grows to twice its size, and the membrane pinches in the middle. The cell can now undergo binary fission. Binary fission is the prokaryote form of asexual reproduction. After the cytoplasm divides, each daughter cell synthesizes a new cell wall, creating two identical cells. Binary fission results in two cells that each contain one copy of the original circular DNA. Prokaryotes are able to multiply rapidly because of their small size. Their DNA takes a short time to replicate, and with a high surface area to volume ratio, they can absorb nutrients quickly. Also, the relative simplicity of the cell's internal organization allows rapid duplication. A bacterium can divide every 20 minutes in optimum conditions. Unchecked, bacterial population growth is exponential, allowing rapid colonization of a favorable environment. Scientists can apply knowledge of prokaryotic cell division in disease prevention and care. Through the use of antibiotics, doctors and scientists can slow or stop the growth of disease-causing bacteria. Some beneficial prokaryotes aid in digestion. Other prokaryotes play a role in the environment and are used for cleanup of toxic waste in bioremediation programs. It's no exaggeration to say that prokaryotes are an essential part of life on Earth.
All right, so we're going to watch it one more time. And again, your job is to click the reply button and post how do bacteria reproduce or how do bacteria make more of themselves.
Prokaryotes are microscopic single-celled organisms, including bacteria and archaea. Prokaryotes are everywhere. If you look, you'd find bacteria on every object around you. One reason bacteria are so ubiquitous is that they multiply very rapidly. How do they multiply so quickly? Let's look more closely at prokaryotic cell division. Prokaryotic cell division is a process by which a cell divides into two or more cells. The process of prokaryotic cell division is called binary fission, which is a simpler process than found in eukaryotes. Recall that prokaryotes differ from eukaryotes due to the lack of a nucleus or cell organelles. Typically, their chromosomes consist of a single loop or circle of DNA. The circular DNA molecule replicates. It then attaches each copy to a different part of the cell membrane. The cell grows to twice its size, and the membrane pinches in the middle. The cell can now undergo binary fission. Binary fission is the prokaryote form of asexual reproduction. After the cytoplasm divides, each daughter cell synthesizes a new cell wall, creating two identical cells. Binary fission results in two cells that each contain one copy of the original circular DNA. Prokaryotes are able to multiply rapidly because of their small size. Their DNA takes a short time to replicate, and with a high surface area to volume ratio, they can absorb nutrients quickly. Also, the relative simplicity of the cell's internal organization allows rapid duplication. A bacterium can divide every 20 minutes in optimum conditions. Unchecked, bacterial population growth is exponential, allowing rapid colonization of a favorable environment. Scientists can apply knowledge of prokaryotic cell division in disease prevention and care. Through the use of antibiotics, doctors and scientists can slow or stop the growth of disease-causing bacteria. Some beneficial prokaryotes aid in digestion. Other prokaryotes play a role in the environment and are used for cleanup of toxic waste in bioremediation programs. It's no exaggeration to say that prokaryotes are an essential part of life on Earth.
All right, so right now I'm going to give you about 5 minutes to answer and describe how bacteria reproduce and how bacteria make more of themselves. Alrighty, so let's look at our answers here and go over them. So, binary fission involves single-celled organisms and they reproduce asexually. So that means they don't need a mate or another organism in order to make offspring. They're a single cell with genetic information just like our cells have, and all they have to do is make an identical copy of the cell and everything inside of it, including the DNA, and they end up with offspring or organisms. So this means that bacteria can divide very easily and very efficiently or quickly. They don't need a mate. They only have to make a copy of one single cell and its components, and so they can do this very quickly, okay? So one bacteria can become hundreds of thousands to millions in a very short amount of time, okay? Relative to how long it takes most organisms to reproduce. So in our next task here, it says we're going to calculate how quickly bacteria divide. And we're going to see, and this is going to kind of build us up to class Thursday, how quickly they can reproduce and quickly we can go from one to many more. So it says here, during this process of binary fission, bacteria copy their genetic information and split in half. One bacteria becomes two bacteria becomes four, and four become 8. It takes most bacteria 20 minutes to divide. It's very quick. Use the time it takes a single bacterium to reproduce to do a quick calculation. So the question you are trying to answer here is, how long would it take to end up with 8 bacteria if you start with 1 and they take 20 minutes to divide each time? So that is the problem I want you to figure out right now. And when you figure it out, you're going to click the reply button and post your answer. How long would it take for us to end up with 8 bacteria if we start with 1 and they take 20 minutes to divide? Okay. So I'm going to give you about 5 to 7 minutes right now to try and figure that out. And again, you're going to click the reply button and post your answer. And then we're going to go over it. As always, let me know in the chat if you have any questions as you're working on it. Alrighty. So your answer should have been it takes 1 hour or 60 minutes. So one bacteria divides into 2, and that takes one set of 20 minutes. Those 2 bacteria each divide into 2, giving us 4 bacteria, and that takes another 20 minutes, so that's 40 minutes so far. Each of the 4 bacteria copy and divide, producing 8, which takes another 20 minutes. So 20 times 3, or 20 plus 20 plus 20, would give us 60 minutes or 1 hour. Okay. Does anyone need or want to ask a question about how I figured that out? Okay. Take the awkward silence as a no. If you do have a question, make sure you drop it in the chat. All right, the last thing we are going to go over is going to be finalizing our plans here for our investigation. So if you recall last week, we are brainstorming different places in the school that we wanted to swab for bacteria. And you had completed a survey to let me know which areas you really wanted me to swab in. Now, normally you would do this, but unfortunately, because of the circumstance, I'm going to be doing the swabbing for us, okay? But you'll still be able to see all the results, and we'll be able to talk about it and test them, okay? So, let's see here. What is our question that we're trying to figure out or answer? What are we trying to figure out or answer in this investigation when I go around to different places and swab them for bacteria? What are we trying to figure out or answer? And what are we trying to figure out or answer by having me go around to different places and swab for bacteria? All right, so which areas of the school are more clean? Which areas are more dirty? Which places in the school can we find bacteria? So someone did put the types of bacteria we could find. Like what bacteria. So it'll be difficult for us to identify the types of bacteria. I will say that a lot of bacteria have their own unique color on their colonies, so we can kind of tell that way. But I think the focus for this one is going to be kind of just looking at which areas have more bacteria. So that could be another way we could word this. Kaylee just sent that to me as well. Which areas of the school have more bacteria and which areas have less? Okay. So remember then in your investigation assignment that you had, it said that we use these materials that are in blue. And so let's go over what we're going to use them for. A sterile cotton swab basically looks like a really long Q-tip, and I'm going to use that to get all in different places and collect nasty samples from the areas.
Are those the same type of swabs that used to test people for COVID?
Basically, yeah. And then I'm going to take my nasty swab and I'm going to smear it across the top of an agar-filled Petri dish. So a Petri dish is just a little plastic dish with a lid, and it's filled with something called agar. Agar is kind of this clear, colorless, almost like Jello-like substance. And kind of like Jello, it's basically water and sugar and proteins. So it has the food that bacteria need to stay alive and healthy. So when I swab or smear my swab across the top of the agar, if there's bacteria, they will have everything they need to grow. I'm going to wear gloves. Permanent marker is going to be used to label the dish so I can remember where each dish's sample came from in the school. And then I put the lid on the dish and I cover it in masking tape and put it in a Ziploc bag to make sure that the bacteria stay inside the dish and don't grow and start to move outside of it. Okay. So you guys then completed a survey telling me where you wanted me to swab, and these were the results. So highest amount of places, door handles at the entrance of the school, my hand, which I find highly offensive, behind the toilet in the bathroom, disgusting. And then it was between the light switch or my keyboard. So I think just because like a light switch, especially a light switch that might be like in the hallway or front office will be touched more, I'm gonna do that one, okay. So we're gonna swab door handle to the entrance of building. Miss Rosema's hand, disrespectful. Behind the toilet in the bathroom, and a light switch. And I'm gonna find one in a hall somewhere that gets used a lot. Then we're gonna swab places that we are assuming are not gonna have as much bacteria. Oh, it does say the bottom of my shoe. Okay, Kaylee, you're right. Kaylee cleared that up. That is a little bit less offensive, isn't it? I do expect my shoe to be dirty. Good looking out, Kaylee. Thank you. I'm gonna lose sleep overnight at everyone thinking that I have the nastiest hands. All right, so areas that have very little bacteria. Cafeteria, right? And I think the logic there, right, was that in a normal year, we would probably swab the cafeteria for lots of bacteria, disgusting, but no one's using it. So our assumption is that it's not gonna have a lot of bacteria. Then we have here the swimming pool. I have lockers, teacher's classroom. A water fountain will be an interesting one. So a lot of these are very close together. So we are definitely going to do the cafeteria. I think the swimming pool will be very interesting, right? Because, you know, swimming pools add chlorine specifically to kill bacteria. So have they kept up with the maintenance of it during a school year when it wasn't used heavily? I think the water fountain will be very interesting. Water fountains in normal school years are very interesting. So I'll be curious to see if it is cleaner since people have not used it. Okay, here. We got some computer lab keyboards. Those could be interesting. Keyboards also can get real funky. So it'll be interesting to see if the computer lab keyboards are better off, knowing that students haven't been using them. Right. So what are some safety precautions I should take when I go around swabbing for bacteria? What are some safety precautions I should take when I go around swabbing for bacteria?
Do it when nobody is in the school.
Why would that be a safety precaution I would want to take?
Because like do it like at the end of the day, but like before they start cleaning stuff, so that you can get like.
Someone take my samples before custodians clean. Okay, that's important for sure. Okay. Should I do anything or wear anything to keep myself safe?
Put a glove on.
Right, wear gloves, wear a mask, good idea. Hazmat suit. Well, you know, we do want to keep in mind, we are normally just in this building all over it, all day, every day, all kinds of normal, right? So gloves for sure. Okay, how many cotton swabs do I want to use? Just one for everywhere? Two. So for my 8 different locations, I want to use 2 cotton swabs. Ah, one for each. So use 1 swab for each sample. Okay, that way we can make sure that the only stuff that's on that cotton swab is whatever I picked up from that area. Okay. Should I put more than 1 sample in a Petri dish? Okay, no, right? So everything we can do to possibly make sure that our samples are isolated and not contaminated from other surfaces will help us really make sense of what we see in our Petri dishes. So use 1 dish for each sample as well. Okay, now data to collect. You had to look up in your homework assignment what bacterial colonies looked like. And you probably went right to Google. And here we have a really good picture of what bacterial colonies look like. If it's water, yes. They came around with bottles of water, but you were sleeping. All right, so here is a Petri dish. The kind of clear substance in it is agar. And these colorful, smooth, almost shiny sometimes looking dots are bacterial colonies. So if there are bacteria in these areas that I swab, when I put them on the Petri dish, we're going to see things like this growth. So when I ask you what data could we collect, we want to think about what actual information could I count or measure and record from each Petri dish. So who has an idea here? What is something that I could count, measure, or record from each of our Petri dishes to help us understand which areas have more bacteria and which areas have less bacteria? What is something that I could count or measure or record from our Petri dishes just by looking at them to help me understand which areas of the school have more bacteria and which areas have less? All right, count the groups of bacteria. So by groups, do you mean the circles? Okay, so one, yeah, count the number of colonies. Remember those circles or groups are called colonies. So I could count the number of colonies per dish, okay? And then measure the groups. So that is one way, right? I could measure the size of all the different bacterial colonies here. But let's have an honest moment. Do any of us, including myself, want to really sit here and measure these super tiny circles to calculate then the surface area of all of these individual circles? No is the answer. But what we can do is something similar. We can estimate about how much of the surface area of the plate is taken up by colonies. So for instance, if I were to look at this plate here, I would estimate that about half or 50% of the plate is covered in colonies, and about half looks like there's open agar space where nothing is growing on it. Okay, so we could do that as well. Or we can estimate the percentage of the plate that is covered in colonies. Okay. So we're going to look at the results of those in a couple weeks. But now we have everything set up so that I can go swab everything. And as we're waiting for me to swab and for the whatever we collect to grow in the dishes, we are going to learn more about how bacteria reproduce and what antibiotics actually do to them. Okay. We are actually done here today, so we are done about 5 minutes early. Remember, your next class is at 11:10. Okay. And if you ever have any questions, make sure you email me. Okay. Have a good afternoon.